guys ready? I think the microphone's on even. That's good. Good morning. Happy Monday. Good to see you. Could you put up the slide of the hair braiding thing there? Oh, you just throw that up there. Okay, now where to go? There's the, it's completely blank. All right, yes. Okay, so there's a special convocation Thursday night as part of the Worship in the Arts Symposium. We have an artist here on campus, and we'll be doing a talk on hair braiding, and I'm planning to attend, and I'm going to see what they can do up here if we can get some braiding done on this. But that'll be an extra convo credit for you Thursday night. Who's next?
The Christmas gala slide. Yeah, just a friendly reminder to come pick up your meal tickets in the Office of Student Life. So the dinner will start at 6 p.m., followed by a jazz concert and a dessert reception. It'll be December 6th. Right, thank you.
Hi guys, I'm Charlie Gibson. I'm on the Worship in the Arts Symposium Committee. I'm a student, student on the committee. So there's a couple things on that. It's at the end of this week, Friday and Saturday. There's a combo for the Worship in the Arts Symposium, so we encourage you to be there. And then at 7 o'clock on Friday as well, here in the FAC, Urban Doxology is going to have a worship session, and we encourage you guys to come out. Urban Doxology, it's a ministry based out of Richmond, Virginia, and they write the soundtrack of reconciliation in a racially diverse and gentrifying neighborhood of Church Hill. They evolved out of urban songwriting internship program. They produce their own music. They offer worship training workshops, worship experiences. They're just a really cool group, and we really encourage you guys to come out and experience and see what it's all about. So once again, Worship in the Arts Symposium, all through Friday and Saturday, with a specific emphasis on the 7 o'clock here in the FAC with the Urban Doxology.
So I'm here to announce about BWC. So this Sunday, we're actually having a pajama day. So you can come to service and after you just roll out of bed and just don't brush your teeth, I guess. And we also have, so it's pajama day this Sunday, and the Sunday after that, we'll have a Christmas party right after service. So please be involved, and it's just been a blessing worshiping as a campus. So thank y'all.
Hi, I'm Ellie Bradley.
And I'm Ronald Gray.
And we're here on behalf of Diversity Council. Tomorrow we have our second annual You Good Roundtable Talk. It's a time for you to come eat some food and just hang out with other peers and talk about your mental health. You can tell us that you're sad or that you're anxious or just that you want to be there and have good conversations. There will be food, great times. Please come hang out with us and talk about your brain. Thanks.
So I'm here from SAC, so I want to tell you guys that we have Pie Night this Wednesday at 9 p.m. You guys chose the pies you guys want, so come and get your piece of pie. Thursday, we have Thresher Cinema, which is Endgame Avengers Endgame. There's going to be pizza and drinks provided, and then soon it's going to come out the winter week schedule, so we have 7 days full of events the week we get back for Thanksgiving break, so watch out for emails and social media on that. Okay, bye.
She had me at pie. And now, here to announce today's speaker is Brad Coleman.
Thank you. So how do I know Autumn? She is a researcher up at the Mayo Clinic. She did not grow up Mennonite, has never been Mennonite, and doesn't really have any Bethel College ties. I'm lucky enough that she joined our family. She married my cousin Ryan, who also works up at the Mayo Clinic. And when I saw what Autumn does for work, I thought of our new mission and vision values that are coming here at Bethel College. I think Autumn does this well. I think that she increases human flourishing to an exciting degree. I think the work that she does and the research that she and her team do has the potential to be life-changing and world-changing. You know, it's wonderful to have you here on campus today, Autumn. It is wonderful every time that Megan and I get to hang out with you and Ryan. We always say, man, we wish they didn't live 9.5 hours away. So thank you for being here today. Thank you for being part of the Bethel College community today. And I am very, very pleased to introduce the wonderful and the brilliant Dr. Autumn Schulze.
Thank you so much. It's such an honor to be here. I'm always incredibly excited to talk about what I do because when I was in college, like you guys, I had never actually heard of oncolytic viruses, and when I heard about them, it was just this defining moment in my life that I'll kind of tell you about. But I'm super excited to tell you about, they're called oncolytic viruses. Onco means cancer, lytic means killing, so it's just a fancy way of saying cancer-killing viruses. I'm an assistant professor of molecular medicine up at Mayo Clinic, and I am going to tell you today sort of a little bit about me. So before I get into sort of what we do, I'm going to tell you about how I got to where I am, how I became an oncolytic virologist, because this is not something that, it's not an answer you get from children when you ask them what they want to be when you grow up. So I had a pretty extraordinary life, you know, if you kind of look at it. I was born in Mason City, Iowa, to this extremely fashion-forward family here. That's my mom and dad, Tom and Echo. And I have one brother. He's an older brother. His name is Matt. And I have to credit Matt with basically my entire career trajectory. I was the younger sister who basically followed him around and thought everything that he did was the most amazing thing in the world, which was incredibly annoying to him. But he took one of the tests that looks at your personality and tells you what you should be when you grow up. But when he was in middle school, and it said biochemist. So he came home to me. I'm in elementary. I have no idea what that is, but I thought that was just the most amazing thing in the world. I have to be a biochemist too. So I kind of went and looked at all of the things involved in that, and so everything I did from then on sort of became more science-oriented. And I was also a huge movie buff. And I watched a lot of science fiction movies, and we watched the movie Outbreak, so I might be getting too old to use this reference. Does anyone know what this movie is? Okay, not quite too old just yet. But basically this movie is about a monkey that gets smuggled into the US. It has a disease, and then it spreads to humans, and then it just causes this huge outbreak, and they bring in these scientists that are dressed in hazmat suits that come and they basically save humanity. And I wanted to be that. I wanted to wear the hazmat suits, I wanted to go in the field, and I wanted to help everyone I possibly could. So much to my poor mother's chagrin, I said, I want to study viruses and go study these things that are hemorrhagic fever and all of this stuff. And so she said, okay, yeah, but I never waned from that. So unfortunately for my mom, that's kind of what I continued to do when I went into college. So I went to the University of Iowa, got my BS in microbiology. And this was probably one of the most defining periods of my life. So about the summer before I went to college, my dad developed an aggressive lymphoma. And about a week before I moved to college, he passed away. And so I had this kind of deciding moment, do I still go to college? Do I still pursue this? And so I did, but I came home every weekend to be with my mom. It had a very significant impact on how my college career actually went. And I always had this sort of lingering, I mean, I love viruses, and that's what I wanted to study, but I always had this lingering being with pre-med students, do I study cancer? You know, I should be helping people like my dad. But I didn't have a passion for it like I did for viruses. So I got my degree in microbiology, and then I went on to graduate school, at the University of Kansas Medical Center. I studied HIV, I went through this, I got my degree there. And I was doing a lot of basic science, and so I was doing the studies that I now know are basically the foundation for all clinical research. So anything that gets into a patient is done by basic scientists. So if you're ever at a time, if you're ever doing research, and you feel like you're not making a difference, you are. You are a pillar that's helping a lot of other people to move up. So I finished my PhD, and I wanted that clinical sort of translation. I wanted to know what that was like. And so I started looking at different places that I knew reputation-wise. So I was looking at St. Jude, Johns Hopkins, and different places like that. But I knew from the experience I had with my dad, so he actually had polycystic kidney disease as well. So basically my entire childhood, we were up and down from the Mayo Clinic. And then when he had lymphoma, we were there quite a bit. And it was always such a really great experience. And so I said, that would be amazing if I could work at Mayo Clinic. And so I started looking at researchers that were there. And I found this guy. His name is Steve Russell. And I watched a YouTube video. He gave a scientific talk on the use of measles virus in order to treat cancer. And hearing that I could use my passion for viruses, which I was basically trying to kill, which I didn't want to do because I love them so much, I could actually use them for good, and I could use that to treat cancer. That was just absolutely the most amazing thing I had ever heard. So that was the quote that got me. At the end of his talk, he said, "I want to cure cancer with viruses." So I immediately sat down, and it probably took me a few days to write this email because I was trying to tell him every possible good thing about myself and really make him want me to work in his lab. So I emailed him, said I really want to work here, got an interview on the phone with him, and he hired me, and so I've been lucky enough to be at the Mayo Clinic ever since. So I moved there in 2011, did my postdoctoral fellowship with him, and then from then I moved up into the assistant professorship role, and now I sort of collaborate with him and a lot of other groups that are there. So that's kind of how I became an oncolytic virologist in a nutshell, but I also like to reiterate the fact that career is not the only thing. There is life that goes along with all of this. So during this period, there are things that get you through. So this is my husband. I'm a huge Halloween buff. I love Halloween, so I love to dress up, and I finally got him to do it. So I met him when we were in graduate school at the University of Kansas, and about a year later, he moved 3,000 miles away to do a postdoc for 3 years. But then he came back and joined me at Mayo Clinic. We got married when we were in graduate school, and now we are the parents of this adorable disaster of a 2-year-old. So I always like to reiterate this. It is completely possible, 1, to be a woman in science, 2, to have a family, and 3, to have a career. You can be a great scientist, you can have a great career, and you can be a great spouse and a great parent. It is 100% possible. Do not let anyone ever tell you that it is not. Okay, so now that's how I got here. Let's go into what we do. So the first thing that we have to kind of understand is, how does cancer develop, and why is it so difficult to treat in a lot of cases? So there is a whole field of study going on how cancer arises, and there's a lot of different reasons. So there's uncontrollable reasons, basically. There's hereditary things, so we have genes that are mutated in our parents that are just passed on to us. There are things in the environment. So viruses themselves, some viruses can actually insert information into your cells that can cause cancer. There are things that we do as individuals that we probably shouldn't. So we can go to tanning beds, we don't wear sunscreen, we are exposed to a lot of different chemicals that can cause cancer. Some people smoke when they shouldn't, and these are all things that can cause damage to our DNA. But there's also the possibility that it's just plain bad luck. You've got billions of cells in your body that they're replicating their DNA every single day, millions and millions of times. So there's a significantly high probability that something's gonna go awry during that process. Your body is incredibly efficient at making sure that doesn't happen, but sometimes it does. And so sometimes it's just bad luck that during just a normal process, you get a cell that has some sort of damage to the DNA that then makes it unable to basically die. So cells will replicate themselves, they'll get old, and they'll say, okay, I'm too old, and they'll die off. But these cells basically lose the ability to do that, and they will just continue and continue to grow at a much more rapid rate than the normal cells. So you'll develop these types of cancers. You'll get cells that mutate even further from that, and so those will be different from the original cells that mutated. So then you've got 2 different populations. You get tumors that will infiltrate into tissues, so they can basically process into these tissues where we can't get them. We can't resect them out with surgery. And so they become very inaccessible to any of the treatments that we would give. They form barriers around themselves, and they basically look like this cell. So your body doesn't normally recognize it as a foreign thing, so it doesn't do anything about it. So if we think about traditional cancer therapies, we've got chemotherapies, radiation, and surgery. Those are kind of the basic standards. These are getting much, much, much better than they used to be, and they do a lot of good. The major problem with a lot of these is that not only do they kill the cancer cells, but they kill your normal cells as well. And a lot of times it's your immune cells, so you sort of lose your defenses. You can't fight off different infections. So ideally, we would like a therapy that not only kills those cancer cells, spares your normal cells, but we also want it to educate your body to say, okay, if this happens again, you take care of it right away so we don't have to go through all of this again. So the question becomes, what can do that? And history tells us that viruses can actually do this. So the oncolytic virus therapy field basically arose through observational studies. there were a lot of cases where, so this is a child who developed Burkitt's lymphoma. This is right when he was diagnosed, and you can see the mass on his right eye. So he had undergone no treatments, but he had contracted a measles virus infection. And over the few weeks, they could see that the mass was going away, and eventually it completely disappeared, and he went into remission. And there were a lot of different cases like this. So there was a couple of lymphomas, and there were a lot of different things where they would get these. They would have subsets of patients that would get these natural infections, and the cancer would go into remission. So it kind of sparked the idea that, okay, well, that makes sense. You've got a virus that's going to go in and kill things. We could just deliver the viruses to these patients and then target the cancer cells. But that's a really hard sell to say that you want to stick a bunch of virus that normally causes really bad things. They're normally thought of as the bad guys. You want to put that into a patient. So I think pretty much anybody in here could think of a virus that does very bad things. You've got influenza. This was the 1918 Spanish flu outbreak that killed millions and millions of people. We've got Ebola outbreaks going on right now. Measles virus is kind of making a comeback. Herpes, poliovirus, all of these types of things. So when you normally think of viruses, you think of these as bad guys. But over the years, we have studied these profusely. So we know so much more about viruses now. We know where they come from. We know how they get in. We know how we transmit them from each other. We know how animals transmit them to us and how we can transmit them to animals. We know specific components of each of these viruses that cause them to kill cells in general. And we know how they interact with our cells. We also know how to combat them in some cases. So we've got lots of vaccines, we've got lots of antiviral drugs, so we have the ability to treat them as well. So with the information that we know, we can now engineer them to be good guys as opposed to bad guys. So there's, I would say, 3 different types of viruses. There's these wild type viruses that we know they can go into normal cells, they can either kill, they can hide in cells, they might not do anything for a while, they can evade your immune system, they can transmit from host to host. But this is all uncontrolled by us. This is all controlled by nature. And then we have attenuated viruses. So attenuated can mean several different things. It can mean a less potent virus, so something where you've taken out an essential gene so it no longer has the ability to kill anything. It can also mean that it has all of the ability that the original wild type had, but somehow you've controlled it, so now it only will replicate, say, in a brain cell or in a muscle cell. And these viruses also can be engineered to alert the immune system as opposed to evading it. So we can engineer them in specific ways to sort of control the infection that's happening. And then there's the inactive guys. These guys over here, they look like a virus, they talk like a virus, but they're not actually a virus because they don't cause a disease of any kind. But what's great about these is that they can deliver cargo. So if I have a gene that's mutated in my body and I want to deliver the correct one, I can basically load it into the shell of a virus, deliver it to a patient, have it get into the cell, and then express that good gene as opposed to the mutated one that's already in my body. And that's called gene therapy, and that's a whole other field of research that's going on with viruses. These guys, so this is essentially what a vaccine, so some vaccines are inactivated viruses, and so they can go in again and alert the immune system to what's going on. So these are what we call oncolytic vaccines because they don't actually cause the lysis of the actual cell, they don't kill it. These we have been using for oncolytic viruses. So these are 2 different types of fields that are ongoing right now, and we're actually trying to combine these 2 together to make our therapies a lot better. Oops. So this is how oncolytic virus therapy works. So essentially we're going to take a virus and we will inject it either directly into the tumor or we will infuse it into the blood. And so it will go and it will infect the tumor, replicate, and kill the cells. So this is the first phase of oncolytic virotherapy. It's basically direct killing of the tumor. The second phase of this, so this is normally not enough to get rid of a lot of cancer. So if you've got large tumor masses, you've got a lot of cancer going on, it's usually not enough because your body wants to fight off the infection. But what happens during this killing is that you'll get a lot of debris released. You've got a virus infection that the immune system is alerted to. So the immune system comes into that tumor, and now you're basically releasing not only virus proteins and things like that, but you're releasing tumor proteins that the immune system normally wouldn't see. So now you're telling the immune system that, hey, this is a bad thing, you should come in here. So your immune system mounts a response to that, and this is the secondary phase of oncolytic virotherapy, where your immune system comes in and basically mops up what's left of the tumor. So now we know we've got these viruses, we can engineer them. what virus do we use? There are millions and millions of viruses. There are viruses in the ocean that we have absolutely no idea what they do or what they are even yet. So the question becomes, what's your favorite virus? And this is a ridiculously small list, let me tell you. But you can probably pick out a lot of different viruses on here. Some of them are extremely bad, and I would likely say that they will never become an oncolytic virus, ever. But the two that I work with in my lab are, I work with picornaviruses. So I work with, Coxsackie virus and then mango virus. So these picornaviruses are one of the smallest viruses known to us at the current time. I like them because they're kind of like the frontliners. They basically go in and they just sort of decimate, and then they go away really fast. So they sort of go and get the job done, and then your immune system clears them. So I really like these guys. Other people really like the big lumberous guys that kind of come in and hang around for a really long time and just sort of, I don't know, some people like those guys. Other groups in in the molecular medicine department work on VSV and measles virus, so I'll talk a lot about today about the studies that Dr. Russell does with measles virus. And these are all the other viruses, at least on this list, that are being pursued as oncolytic viruses right now. So there is a huge field. There's a lot of different people that will tell you this virus is better, this virus is better. But what I love about this is that you've got lots of people basically building an arsenal to take care of the extremely broad field of cancer. So one virus might be great for one cancer, one might be better for another. You could combine them to do an even better job. So how do we make sure these viruses actually just infect the tumor cells and not the normal cells? Well, it turns out that this actually happens sort of naturally. So viruses, they normally will bind to a receptor on the surface of a cell. Tumor cells, so these guys are basically upregulated. They've got lots going on. They want to proliferate. So they, a lot of times, will have many more receptors on the surface than the normal cells do. So just by sheer probability, that virus is going to more easily get into that tumor cell than it will the normal cell. They also have dysfunctional defense mechanisms. So if this is an immune signal, basically it's going to tell this normal cell, it's going to cause a bunch of signaling in it to activate it and block the virus from even entering or replicating in that, and therefore it will save itself. Whereas the tumor cell isn't able to mount this signaling response, so the virus can get in, replicate, and kill this particular cell. The other thing is that these cells are highly proliferative, so they've got lots of nutrients going on. If I'm a virus, I'm a parasite. Viruses can't replicate without a cell. So I want to go to the place that has a bunch of stuff that I need and has it already there. I don't want to wait for the normal cell to actually upregulate it and have to do all this work for it. So I would rather be in this tumor cell. So this actually happens preferentially. There are a lot of studies going on right now to basically engineer other viruses so that we can target them in very different ways, and I can definitely answer any questions more about some of those mechanisms afterward. The other question becomes, okay, so now we've delivered it to the patient. How do we know where it goes? How do we actually know it only infects the tumor cells and not the normal cells? And there's several different ways that we can track viruses. So you can insert genes into these viral genomes, and some of these are reporter genes is what we call them. So they're ways that we can track these viruses. This is one that we use in our group. It's called the sodium iodide supporter, or NIS for short. It's expressed in your thyroid. It takes up iodine and basically is responsible for synthesis of hormones. So what we did was we inserted it into the measles virus genome. So now when the measles virus gets into this tumor cell, it expresses this NIS gene. And then we can deliver radioactive iodine, which will then go into the tumor cell, and we can use special scanners in order to look at that so we can image a person's body and say, okay, is there an infection going on, and where is it actually going on? And we also have special scanners to detect the tumors as opposed to the normal tissue as well, so we can sort of overlay those images and say, is the virus replicating where the tumor's actually at? So the more cells you've got, the higher the signal you've got expressing that particular reporter gene. So these are kind of the early studies that all went on. How do we get, I mean, this is years and years of work in one slide, which I always find very sad. But so we went to the clinic with a lot of these. There was a lot of clinical trials going on, many different viruses, and we've seen a lot of failures, a lot of semi-OKs, and a lot of clinical successes. One of the major clinical successes was in 2015 when the first oncolytic virus was actually approved for therapy in the US. So it's called Imlygic. It's a herpes virus. Pretty sure everybody's heard of herpes virus. What they did was they basically took out some of the genes from the herpes virus to make it safer. So they protect the normal cells. They make sure it only replicates in the tumor cell, but they also inserted a gene that's normally expressed in your body to sort of stimulate the immune system into this particular virus. So they treated metastatic melanoma patients. They did phase 1, 2, and 3 clinical trials, and they found that this has a demonstrable impact on metastatic melanoma, and so it was approved in 2015. So that was huge for us. But there have also been anecdotal successes, which I find incredibly important. So they sort of validate what we are doing and say, this is possible, keep going, figure this out. And one of these things happened during a clinical trial at Mayo Clinic. So this was with measles virus. These are studies that are done by Dr. Russell. This was kind of, I entered when this was starting to go into the clinical trial. So I've been extremely fortunate to sort of view this actually happening in real time. So they're using measles virus. This is an attenuated measles virus. This is the vaccine strain, one of the vaccine strains that we use basically to vaccinate individuals here in the West. Some advantages, and the measles virus is expressing that NIS reporter gene. Some advantages of it are that we can use that NIS to monitor it in our patients. It uses, this particular vaccine uses a receptor that's really highly expressed on tumor cells, so we don't get an infection of our normal cells. And it's also, again, it's the attenuated vaccine, which has already been proven safe. So we've got a lot of safety studies going on already. So this was a trial in multiple myeloma. We gave a single dose, so we did what's called a dose escalation. We gave some patients low doses. If they did all right, it wasn't toxic, we increased the dose. And then if that wasn't toxic, we continued to increase until we reached a point where either it was, we did start to see some toxicities, we didn't want to go any further, or we were seeing sort of a plateau in efficacy. And we were also able to, so the trial basically showed that this virus was safe even at our really ridiculously high amounts of virus that we're delivering here. And we were also able to show with our NIST that we were getting tumor specificity. So this is just sort of an image that we took. So there is a tumor here and a tumor here. I don't have the overlay to kind of show you, but this is pre-treatment. And so this is with our radioactive iodine. This is the bladder, so it's secreted out of the bladder. And so 8 days later, you can see that there's actually signal within the two tumors. This one is highly robust. And so this means that our virus is actually expressing this NIS reporter, so it's replicating in the tumors. So this patient, I'll go into, she was one of the major clinical successes that we have. So she's a 49-year-old woman. She's got relapsing myeloma. She's basically run the gamut of all therapies possible. She's had stem cell transplants, all the drugs that we could possibly give her, and it's coming back. And she's got a lot of cancer going on. She's got cancer in the bone marrow. She's got a lot of plasmacytomas. I'll talk about the one that's on her forehead. So this was basically protruding out of her head, and it was such a member of the family that her kids named it Evan. So we always talk about Evan, the tumor. This is just a bone scan, so you can see it protruding out. You can see it's basically degrading the bone that's in her skull, so it's causing a lot of problems for her. So this is a picture similar. So this is pre-treatment. You can see that this protrusion is here. You can see the layers of the skull that are disforming. And this is post-treatment when it goes back to normal. So Evan completely went away within, I think it was about a week after her therapy. This is just, so these are the cancer cells. These are her normal cells. So you can see her normal cells stay the same, and the cancer cells go away after their treatment. So this was huge. She actually was a complete responder. Every single tumor in her body, we could see some NIS imaging. They were lighting up. All of the tumors went away. She went into complete remission, and she is now 6 years out of therapy. She's off therapies for everything. She's completely in remission, and she's absolutely doing amazing. So this was one of the best stories that we had. It was the first demonstration, basically, that we could just infuse virus into the blood, and we could eliminate cancer throughout the body. So this, I think, tells us that what we are doing is absolutely necessary. It absolutely can work, and we should continue to do it. The problem was it didn't work for everybody. So now we have to figure out what made her special and why did she respond to it. So she refers to herself right now as a unicorn, and so it's our job now to say, all right, we need to remove this title. You are no longer a unicorn. So the lessons we learned from our clinical studies were basically that, so pre-existing immunity may or may not be a problem. We think especially for measles virus, so a lot of people have been vaccinated for measles virus, so they already have defense mechanisms. So as soon as we put that virus in there, your body attacks it and never gets to the tumor. So we know at least for some viruses that's a huge problem. So there's a lot of field study going on how can we sort of bypass that immunity without basically reintroducing a virus that can cause detrimental effects. So we're trying to change the surfaces of the viruses so that their genetic content is basically the same, but they're using a different receptor, or that they're masked from the antibodies that we make to those viruses. My studies basically want to remove the outer shell of the viruses and just deliver the genetic information. So that's kind of the approach that my research takes, and I can either do that just by directly injecting that genetic material, or I can sort of formulate it into nanoparticles. I can also put our viruses into cells. So if I take an immune cell from your body, so your body knows exactly what it is, it's not going to attack it when I put it back, I can basically load it up with a bunch of virus, re-put that cell back in there, and it can go wherever it needs to go without your body recognizing it, and deliver the virus to wherever I need it to go. So those are all different fields of research that are ongoing right now. We also know we don't get a lot of uptake or entry into those tumor cells. Sometimes the virus just can't get to the region of the tumor. Sometimes it doesn't have enough receptor on the surface. So there's a lot of different things that we need to kind of look at to actually deliver these viruses much more efficiently. We have lots of drugs where we can sort of disrupt vasculature. So blood vessels form within these particular tumors. And if we can destruct those blood vessels within the tumor, we can basically starve it out. So it can't get nutrients anymore. So then if the virus is over here taking care of this part, then we can have a drug that's sort of starving this region of it, and then we can get both sections of it to go away. We can also sort of temporarily suppress the immune system just to give the virus a fighting chance. So we can sort of take out some of the antibodies that you've already got. We can just sort of dampen down your initial immune responses, and then we can deliver the virus, let it get in there and replicate, and then basically let your immune system unleash at a later time point, so we can kind of give them a fighting chance. We also know once they get in there, we just basically don't see a lot of replication. So tumors are really good at evading the immune system, but they're also really good at evading viruses sometimes, because they've basically upregulated their defenses against them so that they can't get in. And so basically what we're trying to do is deliver some drugs to these tumors that basically tame them down a little bit and lower the defenses against viruses, so that way the virus can actually get in and do its job. We also know that the immune phase happens, particularly for our patient with multiple myeloma. We know she had a really robust immune response to this tumor. A lot of patients don't actually have that, so we're doing a lot of different engineering tricks with our viruses to encode things within them that will basically increase the immune infiltration, so the T cells getting into the tumor much more efficiently, the body's ability to tell those T cells which tumor cells to target. And we can also encode, so tumor cells have a lot of proteins that are self, but they also have some that the normal cells don't express. Especially, so if it's a hereditary gene, you know that one's mutated. If I could present that to the immune system could say, okay, anybody who's got this mutation I can take out. So if we encode that within the virus, we can get lots and lots of it expressed, whereas the tumor cell normally just expresses very little of it. So if we express a lot of it, then we can actually get the immune system to do its job. And also they have this immune suppressive environment. They don't want the immune system taking care of themselves. So we have to find a way to basically block all those inhibitory signals that the tumors have, and then go in so that we can actually mount an immune response. So there's lots of things that we have learned through these clinical trials that we are now trying to fix. So just in general, what my research is focused on, I'm kind of focused on the direct killing of it. I like the idea of having a more robust ability to kill these cells. And not just during that primary infusion. I want to deliver a virus, have the immune system come up, which is also going to attack the virus, but I want to do multiple infusions of this virus without having the immune system recognize it. So like I said, I'd like to basically eliminate the surface of the viruses, deliver that using nanoparticles. I'm also really focused on expanding patient eligibility. The majority of our studies are in patients over age 18. We don't treat a lot of pediatric patients for obvious reasons. We know that these viruses can be very detrimental. They don't have great immune systems. And so we really want to expand that. So we have to make these viruses much, much safer. So I've taken several different viruses and done a lot of different engineering to make sure that they don't actually replicate in the cells that they would normally replicate in. And then finally, I really have a big focus on cost. So these drugs, a lot of medical treatments are extremely expensive. And so I am invested in improving and simplifying our manufacturing processes so that we can actually make them affordable and accessible to all populations and not just the most advantaged. So the future, basically, of oncolytic viral therapy is they're proven safe. They have a significant impact in most cases, well, in a lot of cases. They can recruit and activate the immune system. But now we need to sort of make them better. And so we're going to try to use them in thoughtful combinations with things. So different viruses with different viruses, viruses with other immune therapies to boost the immune system, viruses with things we've already got, so chemo, radiation, surgery, so that we can lower the doses that we need to do, but still get maximal effects. So less toxicity, more efficacy. But to do this for the viruses, we've got a lot going on, basically understanding host-virus interactions. So like I said, all of the people doing basic science, looking at these host-virus interactions that aren't actually doing the clinic, are the pillars of what we know and what we do. We need to understand, again, why these certain patients, we've got a handful of patients that are complete responders with different viruses, and we need to know why. And then we need to determine the best time to administer this. We're treating patients basically that have no options left because we're trying to introduce something new. So would this be better if it was a frontline therapy? But that takes a lot to get through to say, okay, you've already got this, we know it works, but we also want to introduce this into there to make sure that we're not going to affect anything with our patients and make them not respond to the good therapies. So just looking at different careers, basically, that impact oncolytic viral therapy, I just have some listed up here. If you're me, you basically go through life thinking that you have to go to graduate school, you have to do a postdoctoral fellowship, a second postdoctoral fellowship, then you become a principal investigator. That was basically all I knew. That was the trajectory if you wanted to be in science. That is not true at all. It is what I wanted to do, though, so that was a good thing. So you can become a professor, you can do investigation, you can do research as a principal investigator. We've got research technologists, veterinary technologists. So research technologist, if you graduate with a 4-year degree, you can go straight and do work in the lab and become a technician. You don't actually have to go on and become the lead of the lab. Our students, our postdoctoral fellows are all very important in this. There's all sorts of different types of science that go into this. So I'm a virologist, but I talk every day with chemists, I talk with radiologists, statisticians. I am horrible at stats, but I have to know whether or not I'm actually have a significant impact. So I talk to people who know what they're talking about there. Epidemiologists, so they can basically all of these different things go into what we do. And then there's kind of the people that are outside, I would say, of the scientific sort of realm. There's the journal editorial staff. So editing, there's graphic design and illustration. So all of the pictures that I have put up here today were made by a software called BioRender. There are people that basically know science and they know pathways and cellular things, and so they basically illustrate that. So I can go in and say, I want to have a picture of a cancer cell dying that looks really nice. And they've made it because they understand what I need and they are very good at graphic design. Google engineers, so we've got people that are basically, we've got big data sets for clinical data and basic virology data. And we need some way to sort of centralize that and have it be accessible to a bunch of people and have us understand it. So we have partnered actually with Google, and we're working on this to have these engineers go between so I can understand IT basically, which I don't. We've got imaging specialists, we've got medical liaisons. So sometimes clinicians talking to a basic scientist is horrible, so sometimes we need people that go between those so you can understand what's going on. We've got medical devices, biomedical engineering is extremely important. People that basically coordinate our clinical trials, our administration. If we didn't have people telling us where to go, what to do, when something was going to be ready, everything would be absolutely horrifying. There are people in sales, pharmaceutical sales, or just basically distributing anything that we need, and talking to someone who actually has research experience or knows what they're talking about is much better than talking to someone who's just trying to sell me something. So I would highly recommend, even if you're in the biology career, you can definitely go into this and do a lot of good. Patent law occupations, and so all of these are different types of things, but I would definitely, if you want to go into something and doing research, I definitely encourage you to seek out research opportunities. So no matter what your skill level is, I would say just try to be in a lab to find out if you like it, if it's right for you. Mayo Clinic has several different programs. So we've got undergraduates, so freshmen and sophomores. We've got summer undergraduate fellowships for juniors and seniors. We've got post-bacs and graduate research educations for people who have their degrees but aren't quite sure if they want to go into graduate studies. And there are a lot of institutions across the nation, across the world that have these types of programs. So if you're just trying to figure out what you want to do, I would highly suggest to go and seek out these types of opportunities. This is my email. I'm happy to answer questions for anybody if you ever want to contact me. I love talking about viruses and what I do, so I will take questions with that.
Questions? Here's a young scientist right over here with a question.
Thank you so much for coming here. You got some good weather, which is an improvement.
It's so much nicer here than Minnesota.
I'm sure, yeah.
We also have places in Jacksonville and Arizona. If you hate snow, Mayo Clinic is also there.
You briefly mentioned that certain viruses work better for certain forms of cancers. Why is that? Why, yeah.
We don't know is the question to that, or the answer to that. I think I mean, one's an obvious answer. So if I'm a brain cancer, I've got a brain cancer, a virus that normally replicates in the heart or the skeletal muscle isn't going to infect there. So there's actually studies ongoing where they're using poliovirus. It's kind of a chimeric virus because polio replicates in the brain. But they took out the part that actually replicates in the normal part of the brain and put in a piece of a virus that doesn't normally replicate in brain cells. So it's the common cold virus. So they basically made this sort of combination of the two, and now it can infect the tumor cells, but no longer infects the brain cells. So it was things like that. It's kind of the natural things that happen. We don't know why, and actually, this is a huge thing because different tumors in different regions of the body are completely different from one another. Different regions within the same tumor are completely different from one another. So my virus might actually infect sort of the right side, but not the left side. It might infect the one in my head, but not the one in my leg. So we may actually need combinations of viruses, and we need diagnoses to say which tumors are going to respond to which virus. So it's incredibly complex and great question.
Thank you for coming. So I have a question. So for the woman that was cured, is there anything like identifying between patients that it has helped cure their cancer? Has there been have you found any
We're doing a lot of studies. Initially, we had thought, so she didn't have pre-existing immunity to measles virus, and all of the patients that we saw sort of like partial responses in didn't have pre-existing immunity. So that's why we think that's very important for the measles virus, at least. For her, so we're kind of looking at the immune response because we know that was really, really robust with her. So she says it was the most intense fever she's ever had after the infusion, and then it went away. So we know she was having a really big immune reaction to this. And so we're kind of looking at her immune cells, and we know that she had a really active immune system versus a lot of our other patients, but it also had the ability to be reactivated. So there's a lot of people that have these kind of like a lot of immune cells, but they can't be reactivated to go after the tumor, whereas hers could. So we think it might be more on the immune side, at least for that particular case.
I just wanted to say thank you that you're a really great speaker because I'm not a biologist person and I could totally understand what you were saying. So I just wanted to say thank you.
Fantastic. Also, if you're terrible at public speaking, keep going. You will get better. I really wish I had a video for this. I was horrible at public speaking when I was in college. Just horrible. Absolutely horrendous. And I just kept going. I went to presentation classes and things like that. It gets better.
I would agree. As a teacher educator, I really like your analogy with the wolf and the domestic dog and then the stuffed dog. Other questions?
A question submitted online. Have you worked with breast cancer with your research, and how successful have you been?
So we just started some clinical trials. So the groups that I work with, so the VSV, I believe they're doing measles virus with it as well. So there are clinical trials ongoing right now at Mayo Clinic for breast cancer. We don't have the data set yet to basically say how successful those have been, but we have seen what we think are preliminary data of responses in those. So yeah, it's a huge field for breast cancer as well.
Do you think like genetics and genealogy can kind of help with this research overall in the long term?
Absolutely, 100%. Basically understanding what sort of mutations are there and understanding the human genome and things like that are very important for what we do. And for diagnoses wise, it's going to be a huge help for which viruses can actually attack those specific tumors. So yes, I'm actually just starting to work with a geneticist, so I'm not super great at it, so I'm learning that. But yeah, I think it's going to be extremely important.
Hi, can you talk about the role of a statistician in your work? I was just curious.
So we do, I'm basically a mouse doctor is what I call myself. So a lot of my studies are in animals, and I've cured a lot of mice of cancer. But I don't do a lot of the patient stuff, and so I'm kind of moving between the two. But for patients, when you're doing clinical data, you're trying to get large subsets, so hundreds of patients before you can kind of determine, does this work? Our animal studies, we're having cohorts of 10 animals, so it's a very different type of statistics that are being done for this. And a lot of the statistics class that we take are geared towards big clinical data. So the biostatisticians they have sort of a different view of this, and they're able to better understand that we're not doing these huge data sets, we're doing smaller ones, and so we need different mechanisms to look at whether or not this is significant. There are a lot of different stats tests out there that you can do, and pretty much if you ran every single one of them, you could find one that worked best for you and say, yeah, look, it's significant, but that's not how you should do that. And so having someone there that understands which one needs to be done, how the study needs to be set up before you even conduct it is extremely important for us.
Thank you so much for talking. So you said that you have 2 viruses that you like to use, and you just said that you've cured a lot of animals of cancer. Have you seen patient success using those 2 viruses?
So actually the Coxsackie virus that I work with, so mangovirus is not in clinical patients just yet. It's actually a rodent virus. And so Coxsackie virus is a human pathogen, and it normally replicates in human cells. It doesn't replicate in mice, so it's very difficult to do animal studies with those because it just doesn't do anything. But Coxsackie virus is actually in the clinic. There's a lot of clinical trials, and the studies with that, I would say it's one of the best oncolytic viruses that's out there. There have been a lot of patients who have responded to that in the melanoma cohort. It's actually proven safer than the one that was approved in 2015. So I think that Coxsackie virus is actually one of the best ones to go after. Mangovirus is really nice because it replicates in animal cells, so I can do immune studies with that one that will then sort of translate to what's happening with Coxsackie virus. Yes with the Coxsackie virus, not so much yet with the mangovirus, but hopefully in the future.
Thank you for speaking today. So with the speed of replication of tumors and their rate of mutation, have you ran into the problem of tumors becoming immune to the viruses that they were given, or is that something that you're looking at?
So they kind of, So that's usually a problem that happens with a lot of drugs. You'll get a lot of patients that can't respond to it. It doesn't seem to happen. It depends on the virus, I would say. So there are a lot of viruses. So my virus, if I put it onto cells, within 24 hours, they're all dead. So there's absolutely no time for them to basically develop resistance to that. Some viruses can, like I said, the more lumberous ones, you can sort of develop that, but viruses are very good at mutating and adapting to those environments. So basically they become the resistant ones. So they can evolve themselves to overcome whatever your cell is doing. So it hasn't been a huge problem that we've seen.
It seems like many people who get involved with medicine are profit motivated. What are some of the roadblocks to reducing the cost of this type of treatment?
So it is, I agree, and so this is something I'm very, very adamant about is reducing the cost of these different things. But so to manufacture, to get a virus prep, it can take, for a fast virus, it can take weeks in order to make these, and you have to go through all of these different tests. So we grow these cells, or these viruses in cells, and all of the material from that cell we have to remove by these different purification processes. And then we have to prove that we remove them, so we're not just injecting a bunch of junk into these patients that can possibly cause even more cancer or cause disease and things like that. So the cost comes when you're actually having to do all of these regulatory steps in order to make sure that the product that you're putting in is okay. So what we've been doing actually is, so to make the genetic material, we don't have to use cells. We can basically do it in a test tube. We just put it at a certain temperature, and then we can just put it on a column and purify it out. That can take hours. So reducing the amount of time we're in the facility, the number of people we have, the number of tests we have to run, can reduce that cost down. So I think it's more, so there's a lot of studies kind of going on with that. There are a lot of profit-motivated individuals, and I think avoiding that has probably so far been my strategy. Yep.
That's all the time we have for today. Let's thank Dr. Schultze one more time. Have a great day.
Thank you so much.